Assalamu alaikum. Welcome to Impact Note. In this video, I discuss 4 new journal articles which explain the role of the alpha cell of the pancreas in diabetes. The first list of articles, the alpha cell regulations of the beta cell function. Actually, it is a review articles explaining the new research finding regarding alpha cell. This is the abstract. The islets of Langerhans is a complex endocrine microorgan which present within the pancreas, contain two most abundant endocrine cell type, beta and alpha cell, which is essential for the blood glucose homeostasis. While beta cell produce insulin, the only blood glucose lowering hormone of the body, alpha cell release glucagon, which elevate blood glucose. Under physiological condition, release product of the beta cell inhibit alpha cell function. The alpha cell releases factors that are stimulatory for the beta cell function and increase glucose-stimulated insulin secretion. The aim of this review is to provide a comprehensive overview of the recent research to the regulations of the beta cell function by the alpha cell, focusing on the effect of the alpha cell secreted factors such as glucagon and acetylcholine. My second article is Defect in Pancreas, Alpha Cell Linked to Diabetes. And this is the author name Sang Kim and a study of the Stanford Medicine. This is the abstract. In response to low blood glucose level, pancreatic islet cells from people with diabetes release significantly more of the hormone called glucagon than do islet cells for healthy people, according to the new study by the researchers at Stanford University School of Medicine. Glucagon is produced by the alpha cell in pancreatic islets, while insulin is produced by the beta cell. Defect of insulin output and beta cell has been thought to be the main driver of the diabetes. The current study, however, supports the growing realization that diabetes is likely due to defect in multiple cell types. This investigation supports the idea that diabetes results not just from a defect in pancreatic beta cell, but also in the alpha cell that produce glucagon. This has been suspected, but their study with human islets provide new and unprecedented evidence to support this idea. My second article is Paragon control of the alpha cell glucagon exocytosis and compromise in human type 2 diabetes. This is published in Nature Communications in 20 April 2020, and this is the abstract. Glucagon is released from the Paragon alpha cell to activate pathway that rise blood glucose. Its secretion is regulated by the alpha cell intrinsic glucose sensing and Paragon control through insulin and somatostatin. To understand the inadequately high glucagon level that contribute to hyperglycemia in type 2 diabetes, they analyze granule behavior, exocytosis, and membrane excitability of alpha cell of 68 non-diabetic and 21 diabetic human donor. They report that the exocytosis is moderately reduced in alpha cells of the diabetic donor without changes in voltage-dependent ion current or granule trafficking. Dispersed alpha cells have a non-physiological V-shaped dose response to glucose with maximum exocytosis at hyperglycemia. Within intact islets, hyperglycemia instead inhibit alpha cell exocytosis, but not in the diabetes or when Paragon inhibition by the insulin or somatostatin is blocked. Surface expression of the somatostatin receptor 2 is reduced in diabetics patient and suggesting a mechanism for the observed somatostatin resistance. Thus, elevated glucagon in human A diabetic donor may reflect alpha cell insensitivity to Paragon inhibition at hyperglycemia. My fourth article says alpha cell to beta cell reprogramming, stepping toward a new treatment of the diabetes. This article is published in Cell Stem Cell journal, and this is the abstract. The beta cell replacement strategy hold promises for permanently treated type 1 diabetes. In Cell Stem Cell restore pancreatic beta cell mass and normalize blood glucose in diabetics mass by reprogramming pancreatic alpha cell to beta cell using PDX1 and MAFA expressing adeno-associated virus infused into the pancreatic duct. So this is all about today's video. Thank you for watching, and please subscribe my channel.